So I welcome all of you, and it's a very known audience here, so I don't want to take every name, and if I'm speaking, I think I need to speak every name because most of them are very close to me. So maybe we'll try at the end of the talk. So what I'll try to do is, so I will try to motivate my title, and I will add a few keywords, and then again I try to show the integration, the concept of integration, which happens naturally in biological systems. The concept of integration. Integration means integration of several disciplines like physics, chemistry, mathematics, computation. Everything is integrated in biological system. So we have a brain which is the supercomputer, right? So when we get along, we'll see the integration of disciplines in the natural way. And as a matter of fact, I also represent an institute which is setting up with the idea of integrated sciences. So SCISR stands for Somaya Center for Integrated Science Education and Research. So you'll see I selected a place which has a similar theme which matches my interest and the natural way of doing things. Like my PhD was at the Raman Research Institute. Then I had my first postdoctoral work at the Mechanobiology Institute, Singapore. Sorry. The Mechanobiology Institute, Singapore. Then University of Bordeaux, it's a CNRS Institute of Neurosciences. So then again at the University of Bayreuth, it's in Bavaria, Germany. So I will add one more word to the title, which is function, structure, dynamics, and then function. Okay, so you will see how these terms are very well connected, and I will also give you a perspective of geometry, which is underlying all these concepts. So this is a kind of my research program. I just want to put 3 axes. So as you would, so people who are familiar with the structural engineering, they will see that there are 3 axes. The vertical axis, it's called the heading axis. For any hydrodynamic structure, for example, a ship or an aeroplane, and that axis, the rotation about that axis determines where you are heading. So it is called the heading axis. And the other axis, which is passing through the wings, yeah. So that's called the pitch axis. So that's the kind of problems we want to pitch on. So it will determine any movement along that, determine the takeoff and landing, for example, where you want to land on some problem. And the long axis along the nose of the aeroplane is called the roll axis. It's kind of rolls when the structure of the vehicle rolls. That's a roll axis. So basically, my heading axis along the soft matter problems, and I pitch sometimes at the mechanobiology and biophysics aspects. And I also handle a lot of advanced technologies, especially in the optical imaging. So along the heading axis, I deal with a lot of self-assembled structures. Like for example, as Dr. Jacob was saying, like how self-assembly, things, how they assemble themselves, how life is getting formed, this kind of problems. And we use diffraction as a method to study. Okay, diffraction is an experimental tool to understand the structures. And along with that, we also use microscopy. Both of these are complementary techniques, diffraction and microscopy. And in the first section, I'll show you how diffraction is a solution to problems. And when you come to microscopy, diffraction will become a serious problem. So now we need to make use of all these techniques and create new knowledge and create an appreciation for the working systems and bring the scholarships. So this is the aim. So the second axis, I show you mechanobiology problems, where again, the first section I will show you assembly of DNA, for example, a polymer, a non-complex macromolecule, how it is assembling and forming structures by itself. And these are very special structures. You need not have to do anything other than mixing few things. They form by themselves. And the second part, I am trying to connect you how the same DNA, which is again packed in very, very intelligent structures inside the cell nuclei. That's what you see in the picture there. Those are cells which are shaped. I have constrained the geometry of the cells. So I can make cells rectangle or circle or whatever shape you want. And then we can look at what happens to the nucleus, what happened to the DNA which is inside the cell nucleus, what you see in green. I'll explain those terms. And how this packaging of DNA within a real cell happens. And this packaging is even more wonderful than the first section what you will see. It is much more intelligent because every now and then this packaged DNA not only pack, you constantly unpack, read out information and pack it back. That's what happens in our cell. So then, I will also show you that this packaging and unpacking, how it is so related to the function and disease conditions. Then, for addressing that, you need a lot of technologies, especially now we are living in the era of very advanced technology. Some of them even thought to not to exist because of physical principles. So this is the idea of interdisciplinary research. There was a physical principle which say that you cannot see things below the wavelength of light what you're using. So that's an old story. So now you can see things with light to nanoscale or without any limit. So I am using those technologies now to really image things at the nanoscale. So that's what you're going along the roll axis. So let's get along. So those are some of them. Yeah. So being a physicist, let me start with a very old textbook physics problem. I think the biology people should not get afraid of this slide, but I'll connect to the biological system in a minute or so. So it's a very old electrostatic problem. An infinite line charge with a linear charge density, say lambda. And the question is that what is it that happens at the point P? What is the electric field at that point P? So all the physics teachers know how to solve it. So you use Gauss law, right? So then you encapsulate a surface, a cylindrical surface, and then you say that, okay, integral E dot ds, and you get the electric field 2 lambda by x. So that's a very straightforward solution. And when you look at the dimension, everything is fine. Lambda is charged by length, and lambda by x is charged by length square, and dimension you are fine. So now in a minute I will show you how a dimensional analysis is going to, you are going to be in problem when you look at potentials. So now I want to say a dimensional analysis, the potential, I want to calculate the potential root. So physics should be complete, whatever route you are taking. I want to calculate what the potential is at P. So phi of x, potential, should depend on the linear charge density and the distance. This is an actual thing to put it up. So then you already know that potential has a unit of charge per length. And then you do a dimensional analysis, you get that potential should depend on lambda to the power, the charge density power 1, and it should not depend on distance. So it is an equipotential surface. So now we are in a problem. So the scaling breaks down here. So potential is constant everywhere. And surprisingly, if you take this, the electric field is, again you get funny answers, right? So now what is the way to get rid of this problem? So the problem arises because you are getting contradictory answer now. That's because the real potential is not constant, it's actually a logarithmic function. So we all studied logarithmic analysis. When logarithms are involved, your scaling, your dimension analysis breaks down. This is one classical example of that. So now, I'm saying, if you have an infinite line charge, the potential is logarithmic. So now we will give life to this problem. So this naturally happens. So you can imagine molecules which are very, very long molecules, like polymers. And imagine that these polymers are charged now. So it is very, in the abstract sense, it's very similar to this problem, right? It's a long polymer and it is charged. So the best example what we have is DNA. DNA is a very, very long polymer which is having very good rigidity, and so now the potential due to an infinitely long DNA strand, it is logarithmic. So because of that, so when you do self-assembly studies, this logarithmic potential will play a very crucial role in determining what structures it's going to form when it's reacting with other species. So even before a DNA reacting to other species, let me give you a few examples, like what all kind of structures you can form using self-assembly. So if you have like matchstick kind of molecules with a tail part and a head part, and with the two conditions, the head part like to be in water, the tail part don't want to be in water. If you throw them on water, so you would imagine the head part want to sit in contact with water, the tail want to be away. So this is what happens when you do introduce these molecules. They all sit with their head down, with the seesaw set kind of thing, right? So then when you have a equilibrium, so you have that kind of structures, and more want to get into water, they form spherical aggregates, they can form cylindrical aggregates, they can also form bilayers. So even some of these bilayers, like sheets, they can also fall off and form very closed spherical structures with the water inside and outside. These are called vesicles. So these are very important biological structures. And the point what I'm trying to motivate here is that, so when you have spherical aggregates, so all these structures are very, very important for biological systems. For example, all your vaccines are piece of nucleic acid encapsulated in vesicular structure or something of that sort, right? All the vaccines, that's what you are being injected. So now there is a consensus for DNA vaccines. So we can use some of the structures. So then, again, all the reactions happening in the nucleus is basically DNA compacted by proteins which are charged, which is called histone proteins. The DNA, how it is, a meter long DNA is packed in 20 micron size sphere. That's your nucleus. So that is again very interesting, and there are several things you can show where these self-assembled structures are very, very relevant, and that is the crucial thing to understand when you are working with biological systems. So again, some little bit more physics of this long chain of DNA. So when you have a long chain of DNA, as I said, the potential is logarithmic. And now, if you take DNA mostly in the form of salt, so there will be a phosphate ion group at the rim, and then there is the counter ions like sodium or those kind of ions which is neutralizing this phosphate groups. So it's like when you put salt in water, the sodium ions and chlorine ions, because of thermal energy, they get dissociated, right? They move randomly. So that is happening, the mixing is happening, there's an enormous gain in entropy, right? So the same thing when you take a DNA salt and put it in water, the salt, the sodium part, the sodium ions will try to dissociate and go everywhere in the solution. So thereby you can gain enormous amount of entropy. But whereas electrostatic is concerned, electrostatic want to neutralize things. So they want to keep all the ions with the backbone. The sodium want to pre-bomb the backbone structure of the DNA. So now you can see there is a competition between electrostatics and entropy. So now there are ways of getting into this game and using this competition and tuning structures. That's what we are going to do. So for example, if you take also I'll introduce one or two concepts. So what is called the persistence length? Persistence length is the rough length scale up to which the DNA strand can be imagined to be rigid, straight. Okay. So for a DNA, it's quite large. It's about 500 angstrom. Whereas if you take polymers like polystyrene, it's very, very small. It's for very flexible polymers, the persistent length will be very small. For very large, rigid structure, the persistent length is really large. So now, if you take this very rigid polymer, the DNA gets its rigidity for two reasons. One is it has a double-stranded mechanical rigidity. Other things come from the electrostatics, which has the charges on its rim, basically coming from the phosphate group. So when you want to bend it beyond certain lengths, the repulsion will come into play. So now, for the DNA, there is a concept called counter-ion condensation. So because of the logarithmic potential, all of these counter-ions will not be released. For a spherical aggregate, all will be released. For a flat geometry, everything will be absorbed because of 1 over r square and log r, which in the case of cylinder. This is the term which is competing. So when you have logarithmic potential, what happens is that about 70% of the counter-ion will be released within the backbone of the DNA. So all these ions will be kept with the DNA backbone. So now, DNA has a negative charge. So now, if you introduce an oppositely charged lipid or a surfactant which has a positive charge, so now they will react. The negative charge DNA and the positive charge lipid, they can react and form very interesting structures. And why do they react? Reason number one is, now when the lipid is coming and neutralizing the DNA, all these condensed counter ions can be released. So thereby it can gain enormous amount of entropy, which is driving this transition. So these kind of transitions are called entropy-driven transitions. Okay, because of the gain in counter ion of the entropy. So again, coming back to the self-assembled structures of surfactant molecule, they can again form spherical aggregates or cylindrical aggregates, and they can form very long structures, which again, long cylinders, and whatever description I gave you for the cylinder, it's again holding good for the cylindrical micelles, but with the fact that they are a little more flexible than the original DNA. Okay. So now you are putting these kind of cylinders with the DNA cylinders, they react and form lot of interesting structures. For example, if you put DNA with a bilayer forming lipid, the DNA strands go and pack between the bilayers, as you could see in the first figure. These are the cross-sectional view. They are like the pipes which is kept between the layers. So they can also form inverted structures where the DNA is surrounded by lipid molecules, and they can form a lattice. It's called a hexagonal lattice. And they can also form one type of inverted hexagonal lattice. Then they can also form interconnected hexagonal lattice. That is in the third figure. So this is the cross-sectional view. This is the surfactant micelle, which are cylinders. And the other one is the DNA cylinders, which are cross-sectional view. So now what we do is that we want to get into this game of entropy and electrostatics. So what we do is that we choose a surfactant molecule which has a strongly binding counterion, which is an aromatic core. It's a very simple choice. So, which means that unlike a chloride ion, a bromide ion, this want to be bound to the structure because it's an aromaticity. So now when you use this aromatic counterion, a cascade of phase transition is going to happen because there is a competition between the entropy of binding and the electrostatics. So we study the structures using a technique called small angle X-ray scattering. So when you have, so it's a typical setup, I think most of you might know. So that is the main beam, and you keep the sample somewhere, and if the angle is at 2 theta, so any scattered beam from 2 theta angle, which immediately suggests you a periodicity in your sample given by the wavelength of the ray used, which is lambda, divided by 2 times sine of half this angle, lambda by 2 sine theta. So that is the rule of small angle scattering. And when you have larger and larger structures, you have to see smaller and smaller angles. And the main beam you can actually stop because it does not have any information, but very close to the main beam, the large structure information will be coming. So one need to be careful with that. So we prepared this aromatic counter-ion, that surfactant, and DNA, a few structures, and just by changing the concentration of that, what we find is that it locks into a hexagonal lattice. This is the structure, this is the scattering pattern, diffraction and pattern, and then it goes to a square, which is the green pattern, and it goes to another structure which we could not activate any of the non-lattice. So if it is, say for example, hexagon, these peaks will be in definite ratios like 1, √3, 2. That's the pattern, that's the periodicity, dictates that. And if it is square, it will be 1 to √2 to half, √5. That will be the positional correlation. So what is in between, we could not index to any of the non-lattice. So this was a big puzzle for us. So I did many samples and I could not find anything now. So then it turns out that, so this has a hexagonal lattice has 6 DNA strands around that. That's a packaging. And a square has 4. So the question is that, can we have a structure with 5 DNA around each micelle? So hexagon is 4-4 symmetry, and square has, hexagon has 6-4 symmetry and square has 4-4 symmetry. With 5 DNA around it, you cannot have a 5-fold symmetry. That is crystallographically forbidden. So what is the choice we have? So it's like 6-fold symmetric structure and 4-fold symmetric structure, and can we have a 5-fold? It's not possible, crystallographically forbidden. So one way of getting that is that if I go and pick up DNA from regular intervals, so I pick a DNA from there, I pick a DNA from there, it forms a much bigger lattice, which is a super lattice of the original lattice. Okay. And you would see that every micelle has 5 DNA around it. This is a very strong possibility. So this could be, so what we could not identify could be a hexagonal lattice with a much larger periodicity. This is one thought, just from the experience with the system. So then I went and checked all the scattering patterns, and it immediately fitted to a hexagonal lattice with the √3 times periodicity of the original lattice. So it is very clear that it's what we see, and I've identified, is a hexagonal, it's a super hexagonal lattice of the original lattice. This is like a kind of a eureka moment. So then, but we need to show that this is indeed the kind of packing we have. So we got the lattice. So now we need to show what way exactly it is packed. So when we try to model the system, we can actually calculate the diffraction pattern. One is you put the sample and shine X-ray, but if you have already know the species, the chemical species, you know the electrons in every atom, and you can put in that information, you can actually calculate the diffraction pattern, and then show that what you get is matching with what you actually predict. That's other way of verifying. So we calculated this, then it was not matching to this structure. But we already have the information that this is indeed a super lattice of hexagon. So then there is something called the brute force method, that you are starting with your electron densities, and then you can actually plot all the electron density maps. Suppose you have 5 reflections. Your lattice is fixed, and your phase, the only information you are missing in an X-ray diffraction experiment is your phase information. Then you don't know which location the sample that is coming, the relative phase. So that is a well-known problem in crystallography. It's called the phase problem. It's still unsolved, but you can circumvent that. So this is one of the way by plotting all the electron density maps, then you can identify, because you know what you are putting in. You know what is the electron density of DNA, you know electron density of surfactant, then you can actually figure out, say, electron rich species could be the DNA, and electron deficient is the hydrocarbon chain. So you can see those kind of structures. So then from this, you need to calculate. So all these are equal possibilities, and then you have to calculate for each of that and show that what is the structure. So final structure what we got is something like that. So it's a very fantastic structure. So if you look at this, it's again a hexagonal lattice with two types of micellar neighborhoods. So these are orangish micelles. These are all, for again, these are the cross-sectional view. This is like the pipes sitting perpendicular to the plane of the display. So these, around this orange micelle, you have 6 DNA strands. Whereas, six-fold distribution of the DNA strands. Whereas for the other color, you have again 6, but they are more condensed to the central DNA. So again you have 6. So this has a 3-fold symmetry, that has a 6-fold symmetry of distribution of micelles. That actually, when I calculated the actual intensity, and it's given a wonderful fit to the original structure. So the diffraction pattern, so this was indeed the structure. That's from the modeling part. But now, this may not be completely convincing for anyone. It's everything fitting, but there could be other possibilities. So now, a powerful model can give you predictions. Okay, you make a model and make a prediction. Suppose this is the structure. So look at this one. Suppose all this DNA is closer to the other one. There is no gap. It is actually a normal hexagonal lattice. So which is same as what you found. It's for certain concentration. So in fact, if you take this structure and apply a stress to bring on this, an osmotic stress, you should actually go to the normal hexagonal lattice. This is the prediction which I made in my PhD thesis. It should happen. So then during my visiting scientist position time, I actually did this experiment. And to a wonderful surprise, this structure indeed went to that structure. It's a completely, wonderful thing. The swollen lattice in some way with an osmotic structure, pressure, you can drive that to the original lattice. So then people can ask why it should form this structure. Why so, So one thing is that it is forming at the intermediate concentration of DNA. So what determines that how much of DNA you are putting in to the with the surfactant. So this structure is forming. You have seen the phase diagram. So there is a phase diagram. So in this concentration, when you come down the axis, you are adding more and more DNA. So at low DNA content, it is forming square, and at high DNA content, it is forming hexagon. And when you have more surfactant and more DNA, it is forming the super lattice. So from that phase diagram, you can take clues. And then you can give an explanation why this is forming. So as you could see, these are all nanopores. See the scale. This is in nanometer, right? The lattice parameter is of the order of about 10 nanometer. That will be the rough scale. And this micelle here, it is sitting in a nanopore made by 6 DNAs. So now imagine a pipe sitting in a nanopore. It has a lot of configurational entropy. It can move like a snake. So like these micelles, and in fact these are not rigid structures. These are molecules actually getting into the structure, escaping, getting into. These are also called worm-like micelles. Like there is a lot of dynamics within each structure. So I will tell more dynamics in the coming slides. So that kind of a configurational entropy is what's stabilizing this particular structure. Okay, so this is indeed an intelligent way of packing DNA with just the energy minimization principle. And you need not have to do anything now to make this structure. You look at my phase diagram, take the amount of DNA, take the amount of surfactant, put it in this, you get the structure. Wherever you do this experiment. So it's a solid information. So now we want to look at what kind of more intelligent structures are packed possible with the DNA. Then I was asking this question after my PhD. Then I found the cell nucleus has much more intelligent packaging of DNA. It is not only packed like this, it is constantly unpacked and read out the information. It's called the transcription process. Then I found a lab which is matching my interest, which is the Nuclear Mechanics and Genome Regulation Laboratory. It is run by one of the Professor G.V. Shivashankar. He was also the director of the institute. So then I wrote to him, I'm interested in looking at the DNA structures. So then he gave me the problem of looking how a transcription activity is happening in the cell nucleus. So transcription is an activity which the transcription machineries will go and open up the DNA and then read the information, again make it and pass that information to some other location. And it's a very dynamic process. We want to monitor them making and forming, but on the average, they maintain the rigidity of the cell. It's a highly dynamic structure. I'll show you some of the pictures in the later slide. So what people have found is that if you take a stem cell and put it on substrate of various rigidities, they become different type. If you put on a soft environment, they become neurons. And if you put on very rigid substrate, they become bone cells. You see the connection. Mechanics connected to biology, the differentiation process. And then, what are the experiments? Shivashankar lab had done that time. They took different shaped cells and look at which RNAs produced more. So they found that depending on the geometry, a large number of cytoskeletal muscle filamentous genes are upregulated when you elongate the cell, and when you make the cell rounded up, they produce an inflammatory response, which means that it is regulating that kind of genes. So what I did was, so then we want to look at how this regulation process is happening. So what I did was this one. So I took two extreme cases of this cell geometry. One is the elongated structure, which is a rectangle, and other one is a circular shape where you don't need to, where the cell need not have to lay out internal cables. Short cables will do. So here you have a lot of long actin fibers running across the length of the cell. So now, what we wanted to show that, so now we will introduce, can you see all these red particles? Yeah, so these are monomers of RNA, it's called UTP, uracil triphosphate, with some labeling. Okay, so we did a smart experiment by putting in, with an osmotic pressure, the UTP can go in, and UTP should localize to the place where the transcription is happening, because the UTP is the building block of RNA. So we try to see where the UTP is going and where the particles are moving. As you could see, there are particles into the nucleus and they are moving away. There are a lot of such particles. And when can we capture what biologists have done using RNA, can we see them with a physical imaging that we have tried. So then we quantified a lot of this. Then we hit what is known as the diffraction barrier, because however small is the particle, it could be one molecule of UTP or it could be a collection of UTP, and using confocal microscopy, these are confocal microscopy images, there is no way that you can distinguish them, because all will appear, anything smaller than lambda will appear on the size of the PSF, right, the point first function. That is limited by the resolution of the microscope. So now, So that experiment to capture the dynamics of the UTP compartments of the transcription machineries was not reached its logical destination, but nevertheless that process was very, very interesting. What we found that when you look at the nucleus, the motion of the nucleus itself, it had a very interesting dynamics. Most of the nucleus was centrally positioned inside that rectangular structure, and whereas the other, it's a kind of a random motion. So you can kind of plot out which kind of filaments are really positioning this. So we figured out that this actin cytoskeleton is positioning the nucleus at the center. So then one more very interesting observation we made is that the way in which this nucleus is moving, the diffusive motion, it has very interesting exponents. Like it is doing something called a moving choral motion. It's like the nucleus is not simply diffusing, it is making some choral, local movements, local diffusion, then the whole choral is getting shifted. So you could imagine, it is like the diffusion process of a pack of animals moving in a forest. So there is a diffusion constant for the animal within the group, and there is another diffusion of the whole group of animals within the forest, right? There are two diffusion constants. So as you could see, the rectangular structure has so much choral structure, right? Whereas the other one is more freely moving. So when you plot the diffusion constant, the time-dependent diffusion constant, so the mean square displacement by time, you have to really quantify this. So then you will see that it has a completely different dynamic signatures, these graphs. So one has a a parabolic kind of a curve, and the other one has a more super diffusive regime. So this has a sub-diffusive regime, then it kind of stabilizes, then it changes over to a super diffusive regime. So these are like very important dynamic fingerprints about the motion of the nucleus itself. So now immediately what we could say, this could be a good biomarker, for example, the diffusion itself. For example, if you take a cell which has a deficiency in one of the protein, the nuclear membrane protein called lamin. Lamin is a very critical protein which gives the rigidity of the nucleus, and it's involved in binding of many things. And there is a problem with the lamin protein in many patients. So there is called laminopathy. If you take a lamin deficient nucleus and look at the motion is very different from the normal one. So your diffusion constants shifts up by an order of magnitude, and there are a lot of details there. So then you can also look at the correlation of the nuclear trajectories. This is the dynamics part. And then you can also do a lot of physics with this one. Yeah, that's a moving choral model. You could also see like a particle in a potential well problem. This is actually a nucleus in a mechanical well, right? So you can get all the, like you trap, like in optical tweezer, like you trap particles. You can do, I did a similar, it's a completely new thought. We can do the same kind of analysis as when you trap a particle in a potential well kind of problem. So I did that, and actually I got very interesting, slopes and exponents. So now, coming back to the problem of circumventing the limit what we can actually see. So if you see, this is the kind of place where we are hit by upper limit between a mitochondrion and a virus. It's about 0.2 micron. That's a limit where light can make us see things. So basically, you can do the calculation and you get around 200 nanometer as a workable, limit. So now, this is a picture I recorded 2 years back with optical imaging. This is one of the best pictures in the world right now. This one. You can see the scale bar here, 35 nanometers. And that's the distance between them. This is taken by a technique called DNA paint. Okay, so this is one of the latest variant of super resolution imaging in the field. And before you develop an appreciation for that, I want to, before you very understand this, I want to give you some basics so that you can really appreciate this image. So one of the way of circumventing the diffraction barrier, which is supposed to be near lambda, which is of the lambda by 2, right, where the imaging light fixes your resolution, which we want to break. So this is broken by 3 scientists who got the Nobel Prize in 2014 very recently. This is Eric Bensig, and this is Stefan Hell, and W.E. Morton. So he is a biochemist, and Stefan Hell is a physicist, actually was a physicist working in Germany. When he proposed to break this idea of breaking the diffraction barrier, nobody took him seriously. So he has to vacate his place in Germany and he moved to Finland. So where he actually showed this, it's actually working. So when he got the Nobel Prize, they called him back and he got the position back again. So again, Eric Bensig, he showed the breaking of diffraction barrier of light in a completely different way. So these two are different techniques. So he developed a technique called STED. You would have heard, many of you. It's called the stimulated depletion emission microscopy. And Bensig developed what is known as a PALM, PALM, photo-activated light microscopy. So I will spend a little bit more time on PALM, which is what's more closer to the image what I showed you. So what the essential feature of the photo-activated light microscopy is that so we know that, okay, there is a diffraction limit and you cannot distinguish things which are within 200 nanometer. Okay, but we know that the molecules are much smaller than that. Suppose the molecules are not emitting all at once. We can make molecule emit at our will. Suppose there is a switch. Okay, so you make one molecule emit. Then it will emit a peak. It's like a collection of photons. A lot of photons will come. Then you are looking like a mountain at a distance. So though you don't know how far the mountain, you can exactly point where the peak of the mountain is, right? So you can actually localize to ultimate precision where the peak of the mountain. So exactly what you are doing is you are getting this beam of photons emitted from single molecule, but you will see that only a sparse subset of the molecules are emitting at a given time. That's a technique. So now I made a small cartoon, but it works, yeah. So it is blinking at, as you could see, that the blinks are molecules which are placed at distance, they are emitting. So now, once they emit, you keep collecting the image. So you keep on collecting the image from your sample. So then you accumulate them, then you actually localize them. Where is actually the location? Then you recreate. You collect each position, and that's your image, right? It can be done once you do the experiment. Within 10 minutes, you can create the image. So you can, in fact, create image of the molecule itself at the nanoscale, and you can also track them because it's blinking and going. You can actually track them. So then, that technique required what is known as a photo-activatable protein. That means you need to have a control at what stage the protein should emit. So usually it is done by another way, let's say UV you use, and you shine a small intensity UV laser. Then a small fraction of the molecule will be excited, which will be emitting, which you are collecting. So usually it is done in a turf microscopy. Turf microscopy is like a Brewster angle microscopy. So you are shining the laser from bottom, and at a larger angle, then everything will come down. And by adjusting the angle, you can actually scan the surface with sample very close to the surface. So it's very, very interesting technique where you can get all the information near the surface. It's also possible to scan the bulk. You have the light sheet microscopy. If you want to do that, that's also possible. So now there are many variants of this PALM now in the field. The first attempt was something called a PAINT. It's called point accumulation for imaging for nanoscale topography. So this happened. This has to do with some very interesting dye called the Nile red. Those who are chemists, they will immediately realize this dye. This is a dye which easily get into membranes. So if you want to, mark the bacterial cell walls, all this dye you can actually count how many bacteria which is like in growing colonies. So you can actually count the numbers. They also get into microplastics in bottled water. You have a pinch of small amount of dye, they can actually show you what the microplastics are there. It's quite a valuable dye. So where I am using is this one. When you introduce this dye, if you have a bilayer structure, so in the bilayer you have the hydrocarbon chain. So the moment you put in the dye, it visits the chain region and it gets somewhere and then again goes back. So goes and forward. So whenever it makes a visit, you get a signal and then you can localize that signal and you can actually make a quite a high resolution image of the visit of the dye from that. Basically, if you want to break the resolution, you want precisely localize it. That's the idea. Localize and break the resolution. So then, one of my mentors, so that is a random visit, the dye's visit. So one of my postdoc mentors proposed what is called as a universal PAINT. So that, what he did is, he put some target on the structure what you want to image, and the dyes will really come and bind to the target. So then you're actually making the dye stand there and emit as many photons it can. Thereby, he increased the resolution quite a lot. So it is a much more efficient technique. It's called universal PAINT, and that is any kind of target you can design. So Grigory was my postdoc, my lab head when I was in CMLS. So then, what further can be done? So what we wanted to do is, when it comes to specific binding, if you want some specific molecule to come and bind and stay there and emit, the natural choice is DNA, the base pair, right? So a group in Germany, actually he was Ralph, he was in US that time, he was my collaborator. So what he did is that he made labeled pieces of DNA which is floating in the solution, and this labeled pieces will come and bind to the target. And the target, the counter piece of the DNA, you will attach to the structure of your interest. So whenever it comes and binds your structure, you know that there is something there. That is your protein of interest. So your protein of interest or anything, any species of interest, you put one piece of DNA and the counter piece float in the solution. So whenever it comes, it goes somewhere and come and bind there, then immediately you get a lot of emission. And this binding time scale, you can actually tune depending on how many length of the DNA RNA chain you want. You can have 10 base pairs or 9 base pairs. Okay. There is a lot of flexibility here. So that immediately gave to almost angstrom scale resolution. But the challenge is that the technology existed before I moved into France. So we had a collaboration between, it's actually Bordeaux, it's Bordeaux people say. Bordeaux means, Bord means land and Au means water. So it's a place name, Bordeaux, and Munich. These were twin cities. So there was a CNRS Max Planck, a joint program. So I was the person who was integrating this DNA PAINT with the initial tracking, single protein tracking. So the challenge was to, this existed as isolated techniques, but the challenge is to put them together for real cell, cell problem solving. So that is the real, there are multiple challenges in this. I will not elaborate on all of it, but I can give you the key points. So if you have a target and you put the counter piece, so they will come and bind like that, and then bind. That is all your signal collection, and you are looking through your objective under the dish. It's all happening in a petri dish, and then it's emitting. It's a cartoon. I'll show you the real picture also. So like that, you collect lot of signals, and now you can actually change the sequence. Suppose you have A, T, C, T, T, T or something, you can put another sequence. And you can do the imaging in the same wavelength. Suppose in the conventional technique, you have 10 proteins, you need 10 different colors. That many laser lines you will not have. You cannot do a single experiment. So now here, you can do them sequentially, the same sample, by changing, you can put one type of sequence for one protein, you can put another type of sequence for another protein, you can put n number of proteins, so it's called multiplexing. So a great technique, multiplexing. So you can imagine n number of colors, and n number of proteins in one go. So this technique advantage, no photo bleaching, because you can supply any number of floating target dyes. These are normal dyes, like any of the Atto dyes. So you can, if they are saturated, you can flush them again with a new, no photo bleaching. This is a major problem in biological imaging. Then no chromatic aberration, as I said, the same color can be used with using different strands. Then multiplexing. Then very simple instrumentation. All you need is a inverted microscope in turf geometry, so which you can build if you want. Then it's really quantitative, because you can put a reference target and you know how many times it's going and binding. And if suppose you have a cluster of 3 proteins, because the only thing which determines the binding time is the concentration of the monomers, which is floating, the colored dye. So if you have 3 times more binding, it means that there is 3 protein molecules at that point. So there is several advantages. Then what we did is, we thought, okay, anyway we are working with the DNA, let us make a template with the DNA itself as a target. And we can give, projecting this binding sites on that. So there is a nice technique called the DNA origami. You can make any structures with DNA. So idea is that if you have a circular DNA or RNA, circular RNA, and if you have complementary strands, so what it does is that it will go and find its complementary and bind it. So if you intelligently put the monomers, you just put, just like the self-assembly process, you can fold the circular DNA in any form, pre-decided. You decide that this is what you wanted to form, and you calculate all your monomers and put it there. It can form the structure what you want. This is the idea of DNA origami. It's basically folding this. It's exactly mean like the paper, cutting involved, right? You can form the structures. So you can make like this is called a tunnel origami. So this is done by Ralph in Munich, but we have done So we have done a lot of imaging of this. So this is how it appears. The real imaging is like, can you see this blinking here? So these are the monomers which the dye particles attached to the DNA strands coming and binding to the target. So then now you collect all those spots and intensities and localize them, and then get a super resolution image. So now it's very interesting in our astrophysical world also. Very often when we look at sky, we see just one spot, a star. Okay. So there is a very interesting star called Lyra. Actually, I met this. I'll tell you some of the excitement when you're doing science. So this Lyra is something like Abhijit Nakshatra. So this seems to be one star for naked eye. Okay. So then, if you actually see that star, it's actually called a double-double. It's not even a double-double. It's actually a set of 7 stars. Okay. So appearing as one star. Okay. So you don't have the resolution to distinguish it. But very interestingly, some of the ancient texts, there is a reference to the star. It is, I think in one of the texts, it's called, I am Abhijit among the nakshatras. So this kind of very interesting connections sometimes you encounter. In fact, I found it like, when you are looking at the resolution issues and all that, like very often when you look at stars, it's very natural for us to think how many points are there in the one single dot we are seeing on the sky, right? In the sky. So then I called this one of the structure as, something like, this is our new new Lyra's constellation. Okay, that's what I call, because these Germans have designed this 7 binding site origami. Then I found that this particular star also has 7. So it is totally, they are not aware of it. It's a random design. They have designed so many other types. Because it's a standard technique, you have some standard preparation, you mix whatever you want, then it forms the structure what you want. You can make 7 or 8, does not matter. But for some reason I have selected this 7 binding site one. And for some reason I called it Lyra. Okay. So this is our new Lyra. And then after imaging, this is what you see. Okay. This is actually very similar to stargazing. So this is all that binding you have seen, different binding events. So all that collected points you localize and then create an image. Now here is the interesting thing. So I called it from Wordsworth 10,000. So I had a glance. So it's not even 10,000, much more than that, because you are putting a lot of these structures. Okay. Now you zoom on each of that. Can you see that? So each of that dot, what you see is a structure what you are putting in, right? So each of the dots. So it's a it's not like you would have seen many super resolution images. Nothing of this sort. Okay, it's not there right now in our place. Each of that, when you zoom down, it appears like that. Those single dot. So then you can also design this in various. You can have pads. This is a 4x3 pad. It's actually after a cell has scrolled on that, you can have various type of structures. So now I showed you how to break the diffraction barrier and go beyond the visible range with the light and image much nanoscale thing. So how do we use this in the biological system? Right? What is the advantage? Now it's going to a completely rigid, and it also sort of explains why I was in a neuroscience institute. So as you have seen, like in the earlier slide of mechanobiology, I was controlling the adhesion of the cell, how the cells were controlled in its shape, how it is sticking to a surface. You can make a rectangular type of cell or circular type of cell. And that controlling is happening at the micrometer scale. That's what I know. The size is the cell size is 20, 30, or 100 microns. Now we are controlling those at that. But really, what is being controlled is the adhesion at the nanoscale, right? But that we are not taking into account. But some way it is, buried in that control. So as you would see, like the situation of looking at a satellite image of a city. Right, of a country, you can see all the cities, right? But you don't know the internal, dynamics of the cities. Now you want to increase, and it's very similar to taking an image of a cell. Okay, for some reason, this was taken in France, and that cell looked exactly like the map of France, and this different locations. Okay, these are different cities, and then each city is a really happening place inside the cell. This is called focal adhesion, these structures. And this is like the feet of the cell, like the one cell attached. And the cell has to move, it has to break down all this structure and then take out its other structure, and then it need to reach out to a new structure, and it need to form new adhesions, and again the process will be. It's a completely, you can't imagine, it's beyond imagination, the kind of processes involved in this, it's just limited by our imagination, that kind of process. So in each of this city or this adhesion structure, there is hundreds of proteins participating. For example, you want to look at each of that. So this is a recent structure, 2010. It's again done by one of my colleagues in the previous institute where I was in, mechanobiology institute. He did it as a part of his PhD work. I think he did it in Harvard. So what he did is to elucidate the seam structure. The integrine protein, it is adhering on the surface, then there is a cell membrane. There are a lot of layering of proteins, and it is a very well-defined layered structure. Okay. So in fact, almost all of many of the drug molecules are all targeted at this integrase. It's a very important family of proteins. In fact, cancers, many of the targets are in integrin molecules. It's a very pharmacologically also very important protein. But the significance is much more than that. Now, if you label any of this protein and image the cell, it should look the same. Why should we believe this? Okay, I tested it. So there's two proteins, the same region. You see, I labeled paxillin, and this is another molecule which is much above that layer, which is taline. You see one-to-one correspondence. Okay. In fact, this image is taken by the super resolution DNA paint imaging. It's actually this structure, you can stretch it to the size of this whole wall, and you can see individual protein clusters. So you can do a lot of control. And then the idea is that, can we take this adhesion control in our hand? Okay. So that's the integrin layer. So now, if I alter that layer, to what extent into the interior of the cell that structure gets propagated? So that means, if I'm able to control the adhesion, it can lead to a complete rewiring of the internal, structure of the whole cell, which has a tremendous impact. Right? You have seen what impact it created just by controlling the macro scale. A lot of genes get upregulated, a lot of genes get downregulated, and now if you are able to control the structure at the nano scale, the kind of impact it can create is very, very significant. So this is why. So these are the adhesion kind of connection I moved. Then why I found this Neuroscience Institute to work with, because they were also very much interested in cell adhesion problem. That was the parallel we had. So this, in fact, if you see neurons, for example, neurons are also type of cells, and these are the connections in our brain. And these cells which are making connections, that is called the synapse, which is the real happening point inside our brains. Right? How these structures are changing, dynamically changing. So these are nanoscale protein clusters, if you can control that. So that is the kind of interest the Neuroscience Institute had. But we know how to deal with the adhesion per se to any type of cell. We are doing a general problem, but those people are interested in getting the specific neuroscience route and the targeted proteins and the pharmacological, those kind of things. But we are physicists, that matter. So then, as you said, you can zoom down to the protein scale. Then what we do is that the same origami, what I showed you for imaging those 7 dots, the same thing can be used as a nano template. Okay. And on that template, you can provide ligands for attaching the cellular protein now. And now you can place the ligand wherever you want. And you can control the spacing of the ligand. Suppose this is the ligand, and you can place them whatever place you want, and you can dictate where the protein of the cell should come and attach. So that is the real control of the nanoscale adhesion. So imagine, you would have seen, one of my interest in this is that, I always see, in accidents, in many things, when you have cuts or deformation, many times you transplant tissues, right? Burns, but you don't get the original restoration. So my thought is that if you are able to control the adhesions at the nanoscale, at the very level of the protein itself, the natural scale of biology, then it should be possible. There is nothing that is impossible to recreate. So we could, in fact, this could be a good route for doing that kind of problems, at least to start with. That is a kind of other interest I have, and other than the neurological significance. So then what we did is we implanted this, we designed this origami and put the ligands on that. And then there are a lot of challenges when we overcome that with certain and skip this for the time. So what I did, that we created two lines where the cell is known to attach. Okay, we can create patterns where the cell can attach, and you can, just like the way you made the rectangular cell or circular cell, there are proteins like fibronectin or a family of proteins. So then you introduce your origami, connected with the ligands at the bottom. So now you just introduce the cell on that. So there is a cell which is connecting along that line and along that line. So it's one structure which I showed you. You make this pattern and introduce the protein, and the cell can actually go and make a yoga pose of that, right? So now the cell has adhered only along that line, along that line. But the cell membrane is between this. It's completely stretched. So now here you are giving your origami templates from the bottom. And you want to now, first you want to see whether that area is a very clean region. So you can see the cell, the complete area. Now you can see this region, you start forming adhesive structures. So you need to be very sure at the nano scale. So what you see is, you see all, these are all the single protein tracking images. You can get all the adhesions of the integrin protein along that line and along that line, and here you see significantly less, there is no adhesion. This is at the single molecule level. Okay. So now you can also see the tracks. As I said, tracking. So you see all these tracks are very, localized, whereas some of the tracks, as you could see, long tracks in this region, which means that they are freely moving integrins in the membrane. Okay. So now to this location, you are introducing the ligated templates at the nanoscale. Then let's assume that then you will be able to localize them. And before going that, you also see that you can really recruit, whatever protein you want. Suppose you are not going to even to cell, you just have floating proteins in place. So it's just a control that you can see the underlying origami structures. What you see red is the origami, what you see the green is the actual protein, and the red has, you can image super resolution image, and you can see exact region. If you take a green region, go underneath, you see the exact shape of distribution of the origami. It's a control experiment, and this is a real experiment. So what you do is that as you will image both the proteins in the same color, because this is at a nanoscale. So if you are imagining the color, this could be shifted using color because of the chromatic aberration. So we got rid of that chromatic aberration in this picture because you want to be very precise. There is an origami structure, there is a protein sitting on that. This is what you want to show. So the red dots are coming, the origami structure, the green is coming from the protein of the cell, and you can actually make them sit in the same location. As you could see, there are many co-localization events, and so kind of very, very promising results we got. So then you can also, suppose you doubt, okay, this is not, is it really coming from my visiting of the cellular protein, or is it coming from some random place? So you can actually see the binding events. So you can see, you see many binding events. It's very, very likely that it's coming from the cellular proteins visiting at the same imaging location. So there are a lot of thick ways of cross-checking this. There are for ruling out other possibilities. So yeah. So one of the very since I mentioned the neuroscience connection. So one of the very interesting consequences is that, see, it's pretty much known now that the clusters of protein in the neuronal junctions is in fact controlling our memory. See, in fact, if you knock down some of the proteins, these are the recent, a few years back, this paper appeared when I just completed that project. So if you knock out some kind of protein from an animal, it cannot forget things. There are very nice mouse models, behavioral experiments they have proved for sure. If that particular protein is not there, it remembers the old location forever. So very well controlled ideas are there. So it is pretty much possible to take control of our memory of things at various levels now, and we understand quite a lot about things, though not in an integrated way in humans. So there are many other applications for these techniques. For example, there are cells. Our cells are constantly exposed to osmotic shock. For example, water, right? So there are structures on the cell membranes called caveoli, which is actually taking control of whenever there is an osmotic shock. It produces a lot of chain of events by swelling, all that. So all those structures can be thousands of those structures can be imaged into one shot using some of these techniques, like a palm, SPT palm. Photo-activated light imaging microscopy. So here, this is a distribution of thousands of proteins. The blue is before the osmotic shock, and the orange is after osmotic shock is introduced. So the entire population of the proteins, which are freely diffusing, is shifted. Right? There is a big shift in that. So this is a work I done with the Institute of Curie in Paris. So then it's a very exciting field. There are dynamics at different levels. We have seen dynamics at the protein level. We can go dynamics. Look, we also showed you the dynamics at the cellular and the nucleus level. So nuclear internal particles of the nuclear, the transcription compartments. Now the whole cell level. For example, this is a structure. I made 2 states and put a cancer cell between. So they do single cell dynamics. And you can track and get a lot of information down. A healthy cell versus a cancer cell, they're completely different dynamics. And you can cluster these behavioral dynamics in very distinct group with a very good accuracy. And yeah, they have all this periodicity, and you can also have many patterns, and you can also do real interesting experiments like how multi-state patterns, for example, how the cells are able to figure out where to go, and they are pretty good, maze solvers. Cells are actually very smart because it's a piece of light, right? The smallest piece of light, and in fact. One of this project was funded by Volkswagen. Because if you think, this is the kind of industrial vision some of the companies have, it may not be immediately connected with that. But if you think about most dream car now, like Google is trying to make a self-navigating car, right? So now, if one car is able to navigate by itself, it needs to have thousands of sensors, and you need to figure out where the pits and paths and all that in the road. But a cell does it naturally. You give different kind of structures and pits and create this kind of structure, and it's able to figure out and go wherever it wants. There are many things which is left uncompleted. I thought I'll come back and do some of these things in our ration, but let's see how it goes. That's why I kind of made a transition. So there are some of these things I have written up where they are in some of the work. Some of the colleagues in Yale and MIT, they have written a book chapter, the translational dynamics part in this chapter. It's written by me. So it is macroeconomics, and then there is about the integrin story. It's a very big chapter. Integrin itself. See, people have spent several lives on building this integrin story, and we did a very minuscule of that. But it's in a new era of new technology, so we have an upper hand in seeing the things the way they are. Because earlier, actually, one of the remarkable things that biologists and biochemists have figured out many things without any of these technologies before. Which is an amazing part. Many of the things, and they got pretty good story about this. Some of them are wrong, but But mostly they have got a good picture, and many of us have to re-verify whether those stories really are there or not there. So there are a lot of things need to be done in this kind of areas. And then about compressive load stuff, again we had a very nice, interesting piece of work where you can see with the load, you see a lot of this chromatin getting condensed, and it's actually reversible. As you will see in the nuclear image, things are condensed and it can open up after the load stress is removed. So when you apply a load, what happens then? To protect the DNA damage, the cell is having a natural response to shut down all its genes. That's what we figured out, that the chromatin condensation is actually a response to the cell to protect its genes. So there are a lot of interesting things we figured out by keen observations, which is the I think this is the fun of doing science in some level. So I acknowledge all my previous laboratories, a lot of places I did there. And this is all the Bangalore University teachers who actually guided us in getting to the platform, and Professor Karat, Dr. Chandrasekhar Shetty, Dr. Vijay Kumar, and the whole Radhakrishnan, so many of them. It was a very nice time, and especially Professor Karat has guided people like me to really go further with science. I thank him and used to tag him this occasion as well. And this one is a picture of the microtubules. Actually, you can see this happening in every second of your. Right now, in all your cells, the microtubules are actually making and forming all these filaments. It's an image taken by me. I find it very interesting. So these microtubules are like comets in biology. So they make and form all this. So if you look at the internal structure of the cell, like in the spider, right, a lot of filaments of various thickness, they are making and breaking at all movements. So we are actually dynamics is happening all the time within us, and we are like an average form what we are seeing. So it's close to quantum mechanical regime. There is things are happening in that. Okay, so I would like to share some of the pictures when Professor Karat has visited us in with your permission, sir, to Raman Institute. So this is a picture where Professor Karat came to inaugurate our alumni association, where he was the chief guest that time. That is Dr. Radhakrishnan, C. Raman's son. This is Professor Masoor. Professor Masoor. So this is the former director of Raman Institute. Actually, gave the first alumni. Actually, the alumni association did not take much of a shape, but this was the first event conducted that time. Then Professor Karat talking to some of his friends and not scientists. So this is a kind of, just I can also, my Guru Parampara, Guru Shishya Parampara, and I stopped with my sleep. So this is the kind of route. So Professor Karat is the second generation of student of Raman himself. So I found the third generation, and many of you also know.
So, is it spontaneously?
6.24. Yeah.
It happens spontaneously, or is it forced?
Yeah. It is. You are changing the concentration. It is not happening in the same sample, but what you are doing is that you are mixing different amounts of DNA.
Okay.
You are converting the concentration. Suppose you put a different ratio, they form a different structure. For a given ratio, it forms one type of structure. So, but there is always never 1-to-1 ratio. So there will be a lot of excess DNA sometimes, or there will be excess affected in other concentrations. So they reach a equilibrium.
And you also had shown the dynamics. How a normal cell behaves and how a cancer cell behaves.
Right.
So can you actually control this cancer cell or control the DNA multiple or DNA mutation of cancer cell with the kind of
cancer thing is a very field as I understand. So there are many attempts to cure it. So it's enormously complex. I would say not very near any of that. But to answer your question, one thing is to quantify the dynamics. That can be captured very, very clearly. You can even look at nonlinear dynamics approaches of plotting the phase space trajectory of this motion, and this will show a lot of very, very interesting features. Like one dynamics can be completely separated from the cancer cells versus the normal cells. So that is one aspect of that. Though it looks very simple two selection of geometry, it has a very, very significant, thoughtful design behind this experiment. Number one, these two shapes actually modeled on epithelial to mesenchymal transitions. There are biologists, and the reverse transition. Okay. So what happens is, if you look at how, if cancer cells are originating one part of the body, and in cancer proliferation, the cells detach from that particular place, and they become migratory, and they get into bloodstream and go to another location. Then they do the reverse, okay, and then do again attach. That's how the cancer actually spreads all over the body. Okay. So if you look at this one, this is a strongly attaching type with a lot of fibers, lot of focal adhesions, very, very strongly attaching. And here, this is actually a migratory type. It's actually trying to detach from that, and the moment if the adhesion proteins are less, it will break apart and run. Okay. So this is a, so you, by controlling geometry, so this is the idea of nuclear mechanics and the interplay of upregulating certain genes, downregulating genes. So geometry, we have shown geometry and the rigidity, for example, it's an important knob to tune. If you want to change the migratory behavior of the cell, even the upregulation of certain genes compared to the other, we have a good knob to do that. And if you look at the dynamics of these, even the structures, membrane structures, they're all very different. Like, and if you look at the internal wiring of these cytoskeletal elements, so you will see this nucleus is under a lot of pre-stress because a lot of cables are running on that nucleus. But whereas this is kind of free, it's also sort of reflected in the dynamics, the fingerprint dynamics what I showed you. One had a U type of that diffusion curve, the other had a very different, right, super residual. So there are a lot of this kind of new knowledge what we bring in. All of this will contribute in some way in addressing, how the way we can tune the behavior of, anomalous cells compared to the thing. And even the lamine experiment I showed you, but the nucleus did not have a lamine. Its dynamic is very different. So then one could figure out, like, okay, we need to introduce this upregulate this kind of protein in that cell to make it normal. So what is it that one can do? So, but the problem with the biological system is that when you turn one knob, there are several other 100 knobs, connected, which will be the downstream of one thing. So that is the complexity level we are handling. And sometimes we could be lucky. Yeah, that's the way I would like to. There are no immediate remedies for it.
You explained that ligand comes and which attach to that cells and all. By controlling that, you told that the cuts can be healed. Cuts in the body parts.
Not an immediate claim, but in that direction we can move things.
So, in that direction also I'm telling, in future, made possible to grow the body parts, like hands, legs.
Actually, now one of the hot things happening in the field is that organ on chip, right? So, lab grown organs. So, this one actually you take stem cells. So, what one actually would like to do is that, see, in fact, I showed you some mouse experiments, right? Like, it's not by me, it's published research by someone else. The mouse, they control the neuronal adhesion and all that. So, in fact, one of the things being a neuroscientist, at least exposed to that. So every time they want to do an experiment, they have to kill a mouse. So this is not like that. So we want to do something alternative. We want to find alternatives. So one other thing is that suppose you take stem cells and you have a room now, already you transfer it to robots. You can do that. You can do already. And you can grow now 20 foot size collection of neurons and how it further shapes fully grown brain or a muscle. That's a way to forward from hurting other animals. So it's a nice way to progressing pretty good. Lab grown things are already in the field. But to really grow to the existing shapes and preserve all the features, we need to work it out to provide exact physical, chemical, and environmental use for that. So it's a challenge that I think it is just a technological challenge, not a deep physical calculation.
Now, as I aware, this year Nobel Prize has given for the determination of the structure of the protein by using the AI tool. Now, whether this is the AI tool is also used in this type of research, and what is that impact?
But there is a lot to do. For example, for designing adapted proteins, how to. You can simplify a lot of things. For example, to design the way in which the origami need to fold and give you a structure. Now you can give that information to AI. You say AI, this is the final structure I want. Otherwise, somebody has to sit and write a program and then code it. It's already a technology. You can copy and paste the technology, but you can use AI to structure those kind of things. It's a very limited context. But on top of that, because as I said, if you tune one node with certain inputs, it is tuned another 100 nodes. Those kind of things we can actually pass to AI, because how many correlations you can do and you can do them. So we have other than using AI giving answers to the problems given in a class, we have a very good time to address many of the challenges. But most important thing is that we need to generate the lab data. That AI cannot generate as of now. So once you have a strong primary data, experiment, using your natural intelligence, then that processing, because AI basically you need to feed data. So that part can produce so that will be helpful.
ಅರ್ಥವಾಗುವ ರೀತಿಯಲ್ಲಿ ವಿವರಿಸಿದ ಡಾಕ್ಟರ್ A.V. ರಾಧಾಕೃಷ್ಣನ್ ಅವರಿಗೆ ತಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಧನ್ಯವಾದಗಳು. ಕಾರ್ಯಕ್ರಮ
ಎಲ್ರಿಗೂ ನಮಸ್ಕಾರ. ಟು ಬಿಗಿನ್ ವಿತ್ ಪ್ರೊಫೆಸರ್ ಪ್ರಕಾಶ್ ಕಾರಟ್ ಅವರಿಗೆ ನನ್ನ ಗೌರವವನ್ನು ಸಲ್ಲಿಸ್ತಾ ಇದ್ದೇನೆ. ಎಷ್ಟೋ ಜನ ಪ್ರೊಫೆಸರ್ಸು ಲೆಕ್ಚರರ್ಸು ರಿಟೈರ್ ಆಗಿ ಒಂದು ವಾರದಲ್ಲಿ ಒಂದು ತಿಂಗಳಲ್ಲಿ ಮರೆಯಾಗಿ ಮಾರುತ್ತಾ ಹೋಗ್ತಿರ್ತಾರೆ. ಪ್ರೊಫೆಸರ್ ಕರಾಟ್ ಇರ್ಬೋದು ಅಥವಾ ಪರಿಚಯಿತ ಪಟ್ಟಾಭಿ ಅವರ ಬಾಲಕೃಷ್ಣ ಅಂತ ಪ್ರಬುದ್ಧರು ಜೀವಮಾನ ಪೂರ್ತಿ ನೆನಪಾಗುವುದು ಅವರ ಟೀಚಿಂಗ್ ಇಂದಾಗಿ ಮತ್ತು ಅವರ ಮಾನವೀಯ ಸಂಬಂಧಗಳಿಂದಾಗಿ. ನನಗೆ ಪ್ರಕಾಶ್ ಕಾರಟ್ ಜೊತೆ ಪಾಠ ಕೇಳುವ ಅವಕಾಶ ಇರಲಿಲ್ಲ. ಸ್ವಲ್ಪ ಸಮಯ ರಿಸರ್ಚ್ ಮಾಡ್ತಾ ಇದ್ದೆ. ಆಗ ನಾನು ಬಚ್ಚ ಆಗಿದ್ದೆ. ಆದರೂ ಕೂಡ ಅದನ್ನು ಅವರ ಸಮಕಾಲ ವೃತ್ತಿಯಲ್ಲಿ ನನ್ನ ಟ್ರೀಟ್ ಮಾಡಿದ್ದಾರೆ. ಅಂತವರ ಸಂಖ್ಯೆ ಬೆಳೆಯಲಿ ಅಂತ ಹಾರೈಸ್ತಾ ಇದ್ದೇನೆ. ಇವತ್ತಿನ ಈ ಟಾಕಿಗೆ ಒಂದು ಅತ್ಯಂತ ಒಳ್ಳೆ ಟಾಪಿಕ್ ಅನ್ನು ಆಯ್ಕೆ ಮಾಡಿದ್ದೀರಿ ಮತ್ತು ಅತ್ಯಂತ ಒಳ್ಳೆಯ ಸಮರ್ಥ ವ್ಯಕ್ತಿಯನ್ನ ಆಹ್ವಾನ ಮಾಡಿದ್ದೀರಿ. ಇವರನ್ನ ಆಯ್ಕೆ ಮಾಡಿದ ತಂಡಕ್ಕೆ ನನ್ನ ಅಭಿನಂದನೆ ಸಲ್ಲಿಸ್ತಾ ಇದ್ದೇನೆ. ಜಯಕರ್ ಭಂಡಾರಿ ಅವರನ್ನ ಎವಾಷಸ್ ಕಾಲೇಜಿನ ಹಾಸ್ಟೆಲ್ ಅಲ್ಲಿ ನಾನು ಟೀಚರ್ ಆಗಿದ್ದಾಗ, ಅವರು ಸ್ಟೂಡೆಂಟ್ ಆಗಿದ್ದಾಗ ನೋಡಿದ ನನಗೂ ವಾಪಸ್ಸು ನೋಡುವ ಅವಕಾಶ ದೊರಕಿದೆ. ಅವರು ಹೇಳಿದಾಗೆ ಈ ಕಾಲೇಜಿನ ಹೊರಗಿಂದ ನೋಡುವಾಗ ಯಾವುದೋ ಒಂದು ಪಳೆಯುಳಿಕೆ ಬಿಲ್ಡಿಂಗ್ ಹಾಕಿ ಕಾಣ್ತಾ ಉಂಟು, ಒಳಗೆ ಬಂದಾಗ ವೈಭವದ ಬಿಲ್ಡಿಂಗ್ ಇದೆ ಇಲ್ಲಿ. ಅದಕ್ಕೆ ಕಾರಣ ಪ್ರಾಯಶಃ ಜಯಕರ ಭಂಡಾರಿ ಅವರು ಮತ್ತು ಅವರೆಲ್ಲ ತಂಡದ ಸಮರ್ಥ ಸ್ಟಾಫ್ ಅಂತ ನಾನು ಯೋಚನೆ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಅವರಿಗೆಲ್ಲ ಅಭಿನಂದನೆಗಳು. ಇಲ್ಲಿಯ ಸ್ಟೂಡೆಂಟ್ಸ್ನ ಬಗ್ಗೆ ಬಹಳ ಒಂದು ವಿಶೇಷ ಮಾತನಾಡಬೇಕು. ಆ ಬಂದು ಪ್ರೇಯರ್ ಮಾಡಿದ ಒಂದು ಪ್ರೇಯರು, ಅದೇನು ಅದ್ಭುತ ಹಾಡುಗಾರಿಕರು. ಸಾಧಾರಣ ಸೈನ್ಸ್ ಕಲ್ತುಕೊಂಡು ಆ ಥರ ಹಾಡಬೇಕಾಗಿದ್ರೆ ಪ್ರಾಯಶಃ ಸಂಗೀತ ವಿದ್ವತ್ನವರೆಗೆ ಗೊತ್ತಿರಬಹುದು ನನ್ನ ನಿರೀಕ್ಷೆ. ತುಂಬಾ ಚೆನ್ನಾಗಿತ್ತು. ಅದೇ ತರ ಇಲ್ಲಿ ಸಹಕಾರ ಮಾಡ್ತಾ ಇದ್ದೆ ಸ್ಟೂಡೆಂಟ್ಸ್. ಇಂತಹ ಸ್ಟೂಡೆಂಟ್ಸ್ ನ ತಯಾರು ಮಾಡ್ತಿದ್ರಲ್ಲ, ಅದಕ್ಕಿಂತ ಬೇರೆ ಬಾಕಿ ಏನೋ ಉಂಟು. ಇಷ್ಟೊತ್ತಾದ್ರೂ ಕೂಡ ಯಾವುದೇ ಸದ್ದುಗದ್ದಲಿಲ್ಲದೆ ಏನೋ ಸಮಯ ವಾಚ ನೋಡಿ ತಿಳ್ಕೊಂಡಿದ್ದೀರಿ. ನಿಮ್ಮಂತಹ ಸ್ಟೂಡೆಂಟ್ಸ್ ದೇಶ ದೊಡ್ಡ ಆಸ್ತಿ ಅಂತ ನಾನು ತಿಳ್ಕೊಂಡಿದ್ದೇನೆ. ಕರಾಟ್ ಅಂತವರು ವಿಶೇಷ. ಎಂಡೋಮೆಂಟ್ ಫಂಡ್ ಮಾಡೋದು ಸುಲಭ, ಅದನ್ನು ಕಂಟಿನ್ಯೂ ಮಾಡೋದು ಸುಲಭ ಅಲ್ಲ. ವರ್ಷ ಅದನ್ನು ಕಂಟಿನ್ಯೂ ಮಾಡಬೇಕಾದ್ರೆ ಚಂದ್ರಶೇಖರ್ ಶೆಟ್ಟರಂತಹ ವ್ಯಕ್ತಿ ಬೇಕು. ತಮ್ಮ ಅನಾರೋಗ್ಯವನ್ನು ಕಡೆ ಲೆಕ್ಕಿಸದೆ ಎಷ್ಟೋ ಸಮಯದಿಂದ ಪ್ರಯಾರಿ ಮಾಡಿಕೊಂಡು ಇಂತಹ ಕಾರ್ಯಕ್ರಮ ನಡೆಸ್ತಾ ಇದ್ದಾರೆ. ಚಂದ್ರಶೇಖರ್ ಶೆಟ್ಟರಿಗೆ ವಿಶೇಷ ಫಿಸಿಕ್ಸ್ ಕಲಿಯುವಂತಹ ಸ್ಟೂಡೆಂಟ್ಸ್ ಇದ್ದಾರೆ ಇಲ್ಲಿ. ಹೊರಗಿನ ಜೀವನ ತುಂಬಾ ರೋಚಕ ಆಗಿರೋದಿಲ್ಲ. ಎಲ್ಲಿವರೆಗೆ ಕಾಲೇಜಲ್ಲಿ ಇರ್ತೀರಿ ಅಲ್ಲಿವರೆಗೆ ನಮ್ಗೆ ಅಡ್ರೆಸ್ ಇರ್ತದೆ. ಒಮ್ಮೆ ಹೊರಗೆ ಹೋದ ಮೇಲೆ ಬೇರೆ ತರ ಇರಬೇಕಾಗ್ತದೆ. ಆ ಸವಾಲನ್ನ ಸ್ವೀಕರಿಸಬೇಕು. ಫಿಸಿಕ್ಸ್ ಎಂಡ್ ಆಗೋದು ICS. I stands for innovation, C stands for curiosity, S stands for success. ಇನೋವೇಷನ್ ಮತ್ತು ಕ್ಯೂರಿಯಾಸಿಟಿ ಇದ್ರೆ ನಿಮಗೆ ಸಕ್ಸಸ್ ಬಂದೇ ಬರ್ತದೆ. ನಿಮ್ಮ ಭವಿಷ್ಯ ಉಜ್ವಲವಾಗಲಿ ಅಂತ ಹಾರೈಸಿ ನನ್ನ ಮಾತುಗಳನ್ನು ಮುಗಿಸ್ತೇನೆ. ನಮಸ್ಕಾರ.
I'm here to present a vote of thanks. First of all, I would like to thank Professor A.V. Radhakrishnan, Sai Sir, Somaya Vidyadhar University, Mumbai, for delivering a wonderful talk. Thank you, sir. I would like to thank Chairperson for today's session, our Principal, Professor Karat sir. Thank you, sir. I would like to thank the guest of honor of today's seminar, in whose name this seminar program has been carried out. My teacher also, Professor Prakash. Thank you, sir. I would like to thank another guest of honor, Professor Narayan sir. Thank you, sir. I would see many former professors of Bangalore University, delegates from other colleges, my friends from other colleges. Without naming anyone, I would like to thank all the delegates present here. I would like to thank all the students. Some of them have left because of, I think, train timing. I thank all the students and all the volunteers who have helped us in arranging this function. Thank you, Anand.